Hi everyone, in this lecture we are going to discuss about the instrumentation of fluorimeter. Fluorimeter is a molecular emission spectroscopy. The principle involved here is emission. Here the molecules, that is the fluorescent samples, will absorb the light radiation and they will go from singlet ground state to singlet excited state, and from there they will return to singlet ground state by emitting some radiation, and that is called as fluorescence. So with the help of the spectroscopy we are going to measure the fluorescence produced by the fluorescent samples, and the students are going to perform a role play on the instrument, and the instrument setup involves a light source, primary filter, then sample cell, secondary filter and detector.
In fluorimetry, we measure fluorescence produced by a substance, but for a substance to fluoresce, it must absorb light. That's why light source is very important. The most common light sources, xenon arc lamp, and another one is mercury vapor lamp. In modern instruments, we use lasers, which gives strong lights with high intensity.
Primary filter also called as excited filter. The filter isolates the wavelength and cause to fluorescence to a compound. The filter placed between the sample cell and the light source. The filter based on the function by the UV light sourced by the wavelength.
Sample cell also known as cuvette. It allows only excitation light. Materials used in sample cell are quartz. Quartz length is 1 centimeter. The light only allows from within one side. The source of the sample cell is, does not have scratches and does not have fluorescence itself.
Secondary filters are placed between the sample and detector, sample cell and detector. It is allow the fluorescence emission wavelength at longer length. It block the excitation length. The purpose of the secondary filters are it improve the accuracy and sensitivity, also enhance the signal to noise ratio. Types of secondary filters are band pass filters, longer pass filters, interference filters. Band pass filters detect the fluorophore at narrow wavelength. Longer pass filters detect at longer wavelength. Now, interference filters, this is mostly selective filter and give highly precision, and it is detect the fluorescence at very longer wavelength.
My topic is about detector. Detector is a component of an instrument that can sense and convert physical signals into electrical signals, such as light, sound, radiation. For example, in UV spectrophotometer, the detector sends physical signals and converts into electrical signals, which is appeared as an absorbance. Types of detector: light detector, photo detector, electrochemical detector, radiation detector, ionization detector, etc. Thank you.
The instrumentation setup of fluorimeter was explained clearly by our students, and we can see the light source is very much important for fluorescence, and the light source must produce UV radiation. UV radiation is necessary for the excitation, as UV is the excitation wavelength that is necessary for exciting the fluorescent compounds, and after the light source we must have a primary filter. In case of fluorimeter we need primary filter, but in case of spectrofluorimeter we need primary monochromator, that is excitation monochromator, and the purpose of primary filter is to filter off the visible radiation and allow only the UV light to pass through. And in case of excitation monochromator, they can be used to allow only the excitation wavelength to pass through the monochromator, and after this primary filter or excitation monochromator we will have a sample cell. The sample cell will be made up of either glass or plastic, and the sample cell will have a capacity of around 5 to 10 ml. We can fill 3/4 of our sample into the sample cell, and after filling it we have to keep it in the sample holder to measure the fluorescence produced by our compound. And after the sample cell we will be having a secondary filter or emission monochromator. The purpose of secondary filter is to filter the UV radiation and allow the visible radiation to pass through. Since at 180 degree angles we will be getting both the transmitted radiation as well as the emitted radiations. So since we all know if the radiation is not observed by the sample, it will pass through the sample container towards the other side, and that is called as a transmitted radiation. And along with the transmitted radiation, our compound is also emitting some radiation, and that is the emitted radiation. So that is why the reason we have kept the secondary filter or excitation monochromator at right angles to the sample cell, because at right angles we can measure the excitation wavelengths clearly. So next to the sample cell is secondary filter. The secondary filter's purpose is to allow only the visible light to pass through, because the purpose of this instrument is to measure how much light radiation is emitted by our sample. That is, we are going to measure only the fluorescence, and that fluorescence is a visible radiation. So with the help of secondary filter we can measure the fluorescence, and also with the help of excitation, that is emission monochromator, we can measure the emitted radiation. So that is the purpose of that. Then after that secondary filter we will be having a detector. The detector's sole purpose is to convert all these light signals into electrical signals, and the detectors can be used based on the type of instruments. Their purpose is to measure how much fluorescence is produced by our compound. So that is the whole instrumentation of fluorescence spectroscopy, and it is a molecular emission spectroscopy because the changes are happening at molecular level and also the emission is happening. So this is the simple representation of fluorimetric instrument.